Welcome back. This section of force pathology, this lab video today is going to be on pure culture technique. This is a Petri dish. Okay, this is what modern microbiology labs use to do pure culture work. But how did Petri dishes and agar plates come into being? Well, it goes back to Robert Koch and Koch's postulates. Robert Koch had a pretty big lab. He had a lot of scientists working with him in his laboratory. And back in those days, in the 1880s, they were growing their fungi and bacteria on gelatin, thin films of gelatin spread on microscope slides, and then they would place those microscope slides under a bell jar to allow the fungi or the bacteria to grow on the gelatin plate so they could examine the microscope slide in under the microscope to see the characters of the fungi or to look at the growth of the bacteria. But that method didn't really work, and when it got warm in the summer, they didn't have air conditioning in the lab, and the gelatin would melt. And so one of the people that was trying to work with these cultures was Walter Hesse. And Walter Hesse's wife, Angelina, had been using a substance called agar as a replacement for gelatin in her fruit recipes at home. And so she suggested to her husband that he try agar. It had been used in Japan for a long time, centuries really, as the solidifying agent in jellies and so forth. And so they tried it in the lab, and it worked great. Now, agar comes from red seaweed. It's an extract. It's a polysaccharide. And what's really cool about agar is this stuff melts at about 80 to 100 degrees C, but then when it cools down, it doesn't solidify to about 40 to 50 degrees C. Okay, so you can heat it up to melt it, but then you can cool it down. When it solidifies, then you can work with it. Okay, the Petri dish. Well, this little Petri dish underwent a development in Koch's lab too. Another scientist, Julius Petri, was trying to figure out a different way to grow these organisms because the microscope slide thing just was pretty crude really, and they had a lot of problems with contamination, as you can imagine, because there's all kinds of microbes floating around in dust in the air, and they would land on that gelatin on that slide, and they would grow too. So who knows what the organism of choice was that the researchers were trying to study and what were contaminants. And so Petri came up with the idea of shrinking the bell jar down to a lid and the dish as the bottom, and to start out as a glass Petri dish named after Julius Petri. Okay, and so we used glass for years and years. Even in the lab when I first started out, we had glass Petri plates, and you would sterilize them by putting them in the oven, heat them to about 160 degrees Fahrenheit for about an hour, and this would sterilize them. Okay, and then once they cooled down, you could pour your agar plates. Well, that took a lot of labor to clean those glass Petri dishes, and the expense of labor this day and age, it's just not efficient. So probably about 30 years ago, before the oil embargo made plastic so expensive, disposable Petri dishes came on the scene, and they have been used ever since as a container with which to hold that agar. So why do you need a pure culture of a microorganism? Well, we need it, for instance, if we're trying to follow Koch's postulates for a new disease. We need to have pure cultures of organisms just to study their characteristics under the microscope, to measure spores, their size, their shape, that sort of thing. We need to have pure cultures to be able to store our microbes, our collection of fungi or bacteria that we have isolated plants for further study. So we have culture collections, sometimes of several hundred different isolates of a fungus like Phytophthora in the lab. So we need to have these pure cultures available to actually work and study with fungi and bacteria. And actually, nematodes can also be grown in what's called auxinic culture, pure culture, using agar as a substrate and some other ingredients in there that the nematodes like to grow on. So these agar plates are really quite useful for studying fungi, bacteria, and so forth. So now what we're going to do is we're going to go into the laboratory here and go through the process of making agar, pouring plates, and transferring microbes into pure culture. And